So I think it's 10. Good morning, everyone. I'm really excited to meet you here. My name is Anna Blocki. You can just call me Anna. I'm an assistant professor here at the Chinese U at the School of Biomedical Sciences. And I'm also a member of the Institute for Tissue Engineering and Regenerative Medicine. And today I will be telling you a bit about research and progress being done in tissue engineering and tissue regeneration. I'm quite excited about it because it's one of my favorite topics. So in case you have questions or you want to say something, you can always write into the Dropbox, not in the Dropbox, into the chat. And I will be trying to ask you questions, so please feel free to reply. Let's have a dialogue here. And so you can also definitely speak up. However, in the meantime, because there are quite a lot of people here today, please keep your microphone muted, okay? Because otherwise there will be a lot of background noise. So let's just start. Now, before we go into like the crazy topic of tissue engineering and tissue regeneration, I'm pretty sure that all of you know the Marvel superheroes, and I'm pretty sure that some of you even have like their favorite superhero or like the favorite superpowers. I do as well, right? So one of my favorites actually is Wolverine. So Wolverine is pretty cool in a way that he has amazing healing powers. So even if he sustains major injuries, for example, through like a couple of shot wounds and like his major organs get damaged, he can repair those. That's pretty awesome. On the other hand, there's also Deadpool. Well, he might be a bit of an anti-hero, but what I really like about him is that he was actually able to regrow his own hand. So basically regenerate his hand. So as you can see from these movies and from the superheroes, the idea of good or enhanced healing abilities or even regeneration has been there for a long while. And humankind is actually dreaming to have these superpowers. And in a way, we as tissue engineers and we as researchers working in the field of regenerative medicine, we are kind of chasing this. So now let me tell you why. If you look into the current potential therapeutic approaches which are around here, we obviously have classical pharmaceutical, like here, different types of pills, etc. But if you then think about additional diseases, such as, for example, several forms of cancers or strokes or diabetes, we are able to treat some of the symptoms, but we are not really able to cure the disease with the current standard pharmaceuticals. Or just think about degenerative diseases such as Alzheimer's, Parkinson's, or even osteoarthritis. All of those are there and we can treat the symptoms, sometimes better, sometimes worse, but we are not really able to undo the damage which has been done. We're not able to cure the disease. So pharmaceuticals do have the limitations. Then we can think about prosthetics. I mean, injuries happen and people sometimes need prosthetics, especially for the lower limbs. But just think about the limitations that prosthetics have. So obviously, we are able to restore some of the function, right? But we will not be able to really restore all function of the foot. So the prosthetic will never be the same as the original limb. On the other hand, there are also diseases which lead to major organ failures, etc. And many of our organs are essential to our survival. So in this case, patients actually rely on organ donations and organ transplantations. But I'm sure you're aware that there is a shortage of organs to be donated. And even if the organs are available, they have to match the recipient. So there has to be a donor-recipient match. And even if there is one, there's always a risk of rejection. And even if there's no rejection, the patients that receive these organs have to live with immunosuppressants for the rest of their life, which brings in a lot of other limitations for them. So basically, they will have a decreased quality of life. So, and this is where we as tissue engineers come in, because tissue engineering and regenerative medicine, which we sometimes just call TERM, can be used to treat diseases that cannot be cured with regular pharmaceuticals and provide natural and living functional organs to overcome the need for organ donors or prosthetics. So this is what tissue engineering and regeneration is all about. So if you think about tissue engineering, the approach is pretty simple. We use cells, materials, which we call biomaterials, and various types of signals, which can be biochemical signals, they can be physical signals, to generate tissue-like structures. And I'm sure that some of you actually saw, for example, the Mesenchymal in the news, because it was like a big thing which went all around the world, where a human-shaped ear was grown under the skin of a newt mouse. So here, actually, I mean, it's only the outer shell, right? But this is actually cartilage tissue grown in the shape of a human ear. So this is one of the examples how tissue engineering can work. And the goal of tissue engineering is to provide biological substitutes that can maintain, restore, or improve the function of damaged tissues. So again, why do we need tissue engineering? I just want to show you some numbers. For example, here we have some numbers of the US, and you can see here the lower graph, the dark blue color here, shows the transplants being performed every year from 2000 to 2016. And now the red line shows you how many patients are still waiting for an organ. Now, even given that all of these transplantations were successful, which is not the case, right? There's still a lot of patients for whom there's no organ available. So organ transplantation has been around and pretty successful since the middle of the 20th century. However, the limited donor availability and also all the limitations, such as, for example, the rejection of those transplanted organs, brings about an urgent need to be able to grow tissues for these patients in vitro. And in vitro basically means in the lab. So really grow these tissues in the lab and have them ready for transplantation for the patients who need it. So basically all these patients are waiting. So this is US. I just wanted to also show you some numbers in Hong Kong. So because you could say, well, US, but Hong Kong, right? We are a population over 7 million people. And these are just numbers from the hospital authorities for 2019 and 2020, just 2020 in half year. And you can see that there, for various types of organs with the majority kidneys, there are actually a lot of patients in Hong Kong, so just around the corner, waiting for a donor organ to be able to continue to live their lives. So as such, it is not surprising that the tissue engineering market is actually growing exponentially. And you can see the trend, and we know that this trend will continue. So as such, there is an immense clinical need for tissue engineering and regenerative medicine. And the ultimate goal is really to overcome the need of donor organs. So this is what we strive for. So now this is a question for you. So you can either write into the chat box, or you can also speak up. I'm happy to talk to you. So what do you think is the difference between tissue engineering and regenerative medicine? Don't be shy. I'm not shy. So we have our answers here. So let me just see. So we have here, regenerative medicine is more focused on stem cells. Oh, that's a nice one. So one of them uses stem cells, tissue about creating new stuff, while regenerative medicine is about repairing old stuff. That's actually a really good one. Or tissue engineering is the technique. Yes. So I get. It's awesome that you're already talking about stem cells, because I will be also talking about stem cells. So actually, the answer is that tissue engineering is more about really constructing the tissues in the lab, so in vitro, and then once they are ready, transplant them. Whereas regenerative medicine is to also use the same kind of ingredients, so bioactive agents, scaffolds, which are our biomaterials or cells, and implant them. And then by implanting them, stimulate the endogenous regeneration processes. So basically induce the surrounding tissue to repair. So it's more like a guide. So this is technically the idea. However, and this is something which you have to keep in mind, you cannot really separate the two from each other, because very often we do prepare a tissue-like construct before, but it's in comparison to the proper tissue, it's quite primitive. So it doesn't have all the different cell types. So it does not reflect the tissue 100%. So it's like a premature tissue. And when we transplant it, it has to integrate into the surrounding tissue. So besides that part of it will become the new tissue, it will also interact and communicate with the surrounding tissue and promote integration and also endogenous healing processes. So it's basically more or less a combination. There's not really a strict line to separate the two from each other. That's why very often we use those two terms interchangeably. Thanks so much for the great idea. So that one is also interesting. So who do you think were the first tissue engineers? Or when do you think people were trying to engineer the first tissues? Or how old do you think tissue engineering actually is? So you can just put in years or tell me where you think tissue engineering emerged first. What do you think? So we have here the late 1900s. Okay, that's a good one. So we have late 1900s. Do we have anything younger or older? Again, early 1900s, 1980s, very good. Early 2000s, so some of you 700s, okay. So some of you think it's a very, very new field. Some of you think it has been around for a couple of hundred years, 1800s, 2000s, very nice. Okay, so you can see it differs. So the first idea to create tissues or to engineer tissues has been around for a while. So Marvel was not the first one to come up with this idea. So as you can see here, the Egyptians already had this idea. And so had, for example, also the Celtic people. So this here is a wooden toe, which was found with the mummy. So now this toe was added to the mummy after the person was obviously dead and mummified. So this toe was meant for the afterlife or for the next life, right? But here they already tried to kind of replace lost tissues. And here, although I guess this was very painful for the woman, in the Iron Age, there was a Celtic woman found that had tooth implants. So obviously, these are not proper living tissues, right? So they are non-living materials, they are rather prosthetics. But if you think about it, that these approaches are like 3,000 years old, the idea and the wish to regenerate and replace lost tissues or limbs or organs has been around for a while. So although the first tissue engineering products were introduced in the late 1970s and early 1980s, so this is where we are really talking about living tissue constructs, the term tissue engineering, so the modern term, so what we understand under tissue engineering, was coined in 1987. So indeed, the tissue engineering field, as we know it today, is around like 30 years old, a bit older than this, right? 30, 40 years. So, and now, obviously, there are a lot of organs that we would like to replace. But what do you think was the first tissue engineered organ that made it into clinical practice? So we have a lot of choice. Which organ do you think is it? So we have skin, we have trachea, oh, very nice. Liver, skin, lung, heart, skin, heart. So pancreas, oh, interesting. Okay, yeah, so I think majority of people actually say skin, some say trachea, heart. Oh, heart is a difficult one though. Okay, so indeed, it was the skin. So this was done in 1981, so very early, during when tissue engineering was really still in its baby shoes. We already had our first tissue engineered tissues being applied in the clinics and transplanted. So just to give you an idea of how that worked. So basically, in this case, here patients with severe burn wounds have been treated, and they receive this kind of skin graft. So now, if you look at the skin, which is here, so we have a picture here of a skin with like a wound. It has different layers. So it has this epidermal layer. No, it disappeared. Here it is. So it has this epidermal layer, and it has this dermal layer. So the dermal layer, and then comes muscle, fat, etc. So, but this dermal layer is vascularized, and it's actually not easy to make because of the vasculature inside. But the epidermal layer, which is like the protective layer of the skin, is pretty amazing. So it's composed of keratinocytes, which sits on top of the basal membrane. So they form like a single cell layer. And they are pretty proliferated. That means that they can multiply a lot. So what they do is basically they multiply and move up, and then they form different types of tissue, and then they form the stratum corneum, which is our protective layer. And I'm sure that all of you know that we basically do shed our skin all the time, right? So these guys are pretty busy, right? Because they have to kind of make up for the shed skin all the time. So you can imagine, since we do not run out of skin under normal circumstances, these guys are really potent, right? So, but the problem with those guys is that although they can multiply up very well, their ability to migrate and thus close the wound is limited. So normally they can maybe like reach a length of around 2 centimeters. So if you have a wound larger than that, they will have difficulties to migrate and close. So in case of burn wounds, which normally have larger surface areas, even though the cells could produce sufficient additional cells to close, they're not able to close the distance, right? So having this information, what the researchers and clinicians did is they basically took a small skin biopsy and isolated this keratinocytes. And then they expanded them, so they grew them in the lab until they had sufficient numbers and put them on a gauze. So, and they kind of put it on the gauze that each cell wouldn't be too far away from the other cells so that they don't have to migrate so much. And then they placed the gauze with the cells on the burn wounds. So in this way, each cell didn't have such a big distance to the next cell, and they could actually close, and they could actually regenerate the epidermal layer, so our protective layer of the skin for the patient. So that was a huge advancement, right? So now let's talk a bit how we actually do it. So I told you before, we are using cells, we are using signals, and we are using scaffolds. And I'm just going to give you like an introduction to each of the three things and how we can combine them, starting with scaffolds. So, but in general, the approach to tissue engineering is very similar in most of the cases. So we use a biodegradable material, so material that upon implantation will go away eventually. And we will try to shape this scaffold or this biomaterial into the shape of the tissue or organ that we need, so it really fits nicely. And then we seed it with cells. Sometimes we add some signals to it, so we ensure that the cells do the right thing. And we implant them either in a premature stage, or we mature it in the lab and then we implant it. And then basically we hope that the scaffold will slowly degrade and that the cells and also the surrounding cells will reconstruct a functional organ or tissue. So that's the whole approach. So now let's have a look at the scaffold. And I'm not sure about how many of you know the extracellular matrix. You have to understand I'm a huge fan of the extracellular matrix. So a lot of my research is actually around the extracellular matrix. And I think it's one of the key factors that we have to know and understand to do tissue engineering. So what you can see here is this is a cell membrane. So everything below here is cell. And here we have the cell receptors. And the cell receptors actually bind to the extracellular matrix. So the extracellular matrix is everything in the tissue that is not cell. So you can see that there are two major types of components. There are fibrous proteins, so fibers. So here you can see these are collagens, for example. And then we have glycosaminoglycans, which are these brush-like structures in between the fibers. So these are like the two major components. Now, the fibrous proteins kind of, because they can form a net or a network, they form the backbone around the cells, and they provide the stability and the shape, right? So every tissue, every connective tissue has these extracellular matrix because this is where the cells sit in. So it's like kind of their scaffold. Then we have the glycosaminoglycan-based components, and you can think about them as very long, negatively charged, huge sugar chains. So actually, all of us are pretty sweet because we have a lot of sugars inside. So all of these are sugars, and they have a lot of negative charge. And that's important because if something is negatively charged, it will draw a lot of water, and it will also draw a lot of cations. So in a way, by drawing in the water, we make sure that all our tissues are really hydrated, because otherwise we will dry up pretty fast. And because these glycosaminoglycans hold such a huge amount of water, our tissues have like hydrogel-like properties. So the extracellular matrix around the cell has hydrogel-like properties, and the huge water content also is perfect for diffusion, etc., which we need in order for all the cells to receive sufficient nutrients, etc. So now, extracellular matrix. So what we are trying to do when we create scaffolds is to actually mimic this scaffold which we see in the tissue, which is the extracellular matrix. And for this, we can use synthetic polymers to kind of mimic the networks of the fibrillar proteins in the ECM. We can use natural components, which can either be, again, fibers, or they can actually be also sugars. So those can be derived from tissues themselves. Like, for example, we can actually extract them. Or they can also be derived from other natural sources, like, for example, seaweed is a very common one. In many ways, when we make scaffolds, we actually combine those two. So we use some synthetic components, we use some natural components. Now, the necessary thing for this scaffold in general is that they are porous. Do you have an idea why? Why do they need to be porous? Why can we not just have like a solid block? So we have a larger surface area. Yes, Felix, that's really good. That's true. That's one of the things. Can you think of other reasons? To absorb shock, diffusion. The shock will, I'll come back to that. Diffusion is a big thing, yes. It can withstand weight of the person, flexibility. Here we go. So that things can grow inside. Yes. So things like, for example, shock absorbance will be more, or to sustain weight of the person will be more depending on the actual mechanical properties of the scaffolds. And then we are able to make very tough and hard scaffolds, or we can make very soft and jelly scaffolds. So all is possible. But one thing is that whatever we do, eventually we'll try to put it into a patient. So we'll try to implant that. And in order to do that, we need it to integrate with the surrounding tissue. And for this, we actually need the pores because we need the cells on the surrounding tissue to grow inside. If we have like a smooth, solid area, this will be very difficult, okay? And in addition, as I told you, we always use scaffolds, signals, and cells, right? And the cells which we see, most of the time we don't want only on the outer surface, but we also want inside the scaffold. So the pores allow them to actually migrate in. Okay. So now, what kind of scaffolds are there? So there are obviously a lot of different scaffolds. So I just wanted to classify them for you, so we get an interesting overview. So we can have pre-made scaffolds where we basically just make porous scaffold first, and then we seed them with cells. The cells migrate in, and then we implant. Or, and that's also a very neat approach, is we try to use extracellular matrix directly. So we use tissue, and we can use this tissue from animals or also from human cadavers. And we basically decellularize it. That means that we remove all the cells so that only the extracellular matrix of the tissue is left. And you can think that this is as similar as it gets to the actual extracellular matrix. Then we seed cells on it and we implant. Or, and that's also a very interesting approach, we use cells. And if you think about it, the extracellular matrix which you find in tissue is cell-made, right? It doesn't come from nowhere. It's actually the matrix which is made by the cells. And we can actually ask the cells also to do that in the lab. So when we grow cells in the lab, they make their own extracellular matrix. And then, for example, because most of the cell culture is done in 2D, so we get like this flat sheets of cells with extracellular matrix, we start stacking them, and in this way grow tissues. Or additionally, I have told you the extracellular matrix has actually hydrogel properties. We can make hydrogels from a lot of different points of view. And then when we make hydrogels, we can also encapsulate cells within the hydrogels and implant. So in the way, it's always scaffold, get the cells inside, and then implant. And in this way, implanted into various defective areas, which can be bone, cartilage, meniscus, or heart here, for example. So now I just want to tell you about some of the techniques we use for that. So one of them is electrospinning. So electrospinning is quite amazing, as we apply high voltages to polymer solutions. And these voltages then enable that we can overcome the surface tension of the polymer solution so that we can make very thin fibers. So these fibers can be even in a nanometer range. So really the thickness of single protein strengths, which we find in the ECM. So you can see this fibrillar structure is very similar to what we see in the extracellular matrix. Or we can do 3D printing. And the great thing about 3D printing is, for example, that we can use 3D scans from actual patients and really get the shape and the architecture needed for this specific patient. So we can do a very well-fitted replacement. Or, as mentioned, we can use hydrogels, jellies, okay? So agar-agar is a jelly, so we can use similar. And in this way, because hydrogels are basically a polymer network with a lot of water, so it works in the same way that it draws a lot of water in, so it's 98% water. We can easily put cells inside, and the cells will survive because all the water content enables sufficient diffusion, so they will not run out of nutrients or oxygen. Or, and this is one of the most recent developments, we can combine these two. So we can combine hydrogels that contain cells with printing, and we end up with 3D bioprinting, where we use those hydrogels and print them into the shape of the organ we want. Okay, so this is very neat because we can already put different type of cell. We can print different cell types and different hydrogels into the structure of the tissue that we want. Or, and this is also something I have already mentioned before, we can use whole tissue. So this is a red heart, and we can remove the cells. So you can see here, there are still a lot of cells, a lot of blood vessels, and we can use different techniques of washing them. And in the end, we just end up with the scaffold with normal cells inside. And this can be reseeded with cells and then implanted. So after we covered the topic of scaffolds, let's talk about our choice of cells. And it's good that you already mentioned stem cells, because stem cells are the choice of tissue engineers. Why is that? Well, it is because, I'm sorry, it's because they can self-renew. That basically means that they can multiply a lot, and they give rise to other more specialized cell types. So in a way, stem cells are the body's raw materials, and cells from these stem cells are then cells with specialized functions. Okay, so basically this is our reserve of cells which give rise to cells with specialized functions. And I just want to give you 2 examples of stem cells that I'm sure you all have heard about or know. So one of them are hematopoietic stem cells. And those cells are pretty busy, and they're because they generate all the cells in our blood, including all cells from our immune system, which is the adaptive immune system, which is the innate immune system. They also generate our red blood cells, which are called erythrocytes, and megakaryocytes, which produce our platelets, which are responsible for blood coagulation. Pretty important, right? So each day, about 3 billion cells are replaced. So these cells have to do this replacement, right? So one example, for example, for our red blood cells, those have an average lifespan of 120 days, and after then they are cleared out. So approximately 5 million red blood cells are removed from our circulation every second. And those stem cells are sitting in the bone marrow and making sure that we do not run out. So now for us tissue engineers, it's actually the mesenchymal stem cells, or shortly MSCs, this is how we call them, that are of immense interest. So mesenchymal stem cells can be found in almost every of our tissues, including vascular tissue, but also avascular tissues such as cartilage. And they can give rise to various different specialized cell types, including bone, cartilage, fat, muscle, tendon, or just the cells which form the connective tissue. So you can see that they can give rise to a lot of special cell types, and by using these cells, we can generate tissues of all these different types. At the same time, they do proliferate quite well, so they can self-renew. So they're a really good source to generate a lot of different tissues. Now, they also have another amazing ability. They can modulate our immune system. So now you would say, well, okay, fine. Well, but we are here talking about tissue engineering, regeneration. We do not talk about infections. Because most people, when they think about our immune system, they think, well, fighting infections. But our immune system is doing so much more. So it basically acts like a traffic police in our body, and it kind of guides and directs a lot of biological processes, including healing and regeneration. And in a way, this processes from the immune system, like for example, the inflammatory response, which also occurs in the absence of an infection, are necessary for healing and regeneration. So if we inhibit inflammation in the beginning of the healing process, healing will not happen. If the inflammation is prolonged, so chronic, which happens in a lot of various chronic diseases such as osteoarthritis, for example, healing will not complete. So you can see that the inflammatory response, which is performed by cells from our immune system, has to be very well directed. And sometimes in the case of diseases, it's basically dysregulated. And this is where mesenchymal stem cells also come in, because they can modulate the immune cells in a way that the inflammatory responses and the immune cells taking part in this inflammatory responses will promote healing and regeneration. So indirectly, they also advise our endogenous systems to promote healing and regeneration. And this makes them really, really potent for regenerative medicine. So now I've been talking about stem cells, which we call adult stem cells, because we can find them in our body. There is another type of stem cells I would like to point out, and this is the embryonic stem cell. So the embryonic stem cell is something that we do not find in ourselves after birth, but these are stem cells which occur very early during development. So after fertilization of the oocyte, a morula will form, and subsequently a blastocyst. And the blastocyst has like two main structures. It has like an out. Excuse me. No. Let me just go back to. Okay, I think it's right. So it has an outer structure with and then it has like this. This thing is really sensitive. So, and then it has these cells here in the center. Okay, this group of cells, and which is called the inner cell mass. Now, the inner cell mass is the group of cells that will give rise to the whole embryo. Okay, which basically means that these cells have the potential to differentiate into every cell type in our body, and that's really strong. And at the same time, if you think about that, we start with a small cell cluster, but at birth we have like a 3-kilogram baby. Okay, so you can think about it, that these cells really have a good proliferation potential. So they can, their ability to multiply is almost limitless. Okay, and in such way, because we can get these cells out of the blastocyst and we can culture them, we basically have an unlimited of source that can give rise to all cells of the human body. Now, of course, this process will destroy the blastocyst. So we will destroy and basically no embryo, no baby will form out of it. So you can imagine that there are huge ethical concerns. Plus, these cells do have a certain genetic makeup, and whatever tissues we would generate from those, they might not match our patients, right? So there might be, again, a donor-recipient mismatch. So it's not the most perfect solution. However, we found a way how to make cells which are very similar to this embryonic stem cells. For this, we can use fibroblast cells. So these are cells that we can also find in our skin, but they're not keratinocytes, but they're sitting in the dermis below. And we can isolate them by a very, very small biopsy. And then we forcefully express genes inside this fibroblast cells, which are genes that are normally expressed by embryonic stem cells. And by forcefully expressing these genes, so it's a genetic modification, we can reprogram the cells into induced pluripotent stem cells. And these induced pluripotent stem cells, or iPS cells, are very, very similar, if not identical, to embryonic stem cells. So they can proliferate a lot, and they can give rise to all different types of tissues. So now the problem with cells in general that have this unlimited proliferation potential is that they can actually grow tumors, right? So there's a risk if we transplant the cells and they are not properly differentiated that there might be a tumor formation. And another thing is to really push those cells into exactly the cell type we want. So now biochemical signals, or signals in general. And then we have various types of signals, such as, for example, growth factor. So here I have an example for you, which is the vascular endothelial growth factor, or shortly VEGF. Now, these growth factors are secreted by cells, and they can diffuse to other cells and bind the receptor of the cell membrane. They induce a lot of different signaling pathways and then change the gene expression of the cell to, for example, induce angiogenesis. So VEGF induces angiogenesis, which means the sprouting of new blood vessels from pre-existing ones. And if you think about regeneration and wound healing, growing of new blood vessels is pretty essential. So VEGF is a very potent factor. But that's not the only one. We also have mechanical signals. And this, for example, can be transduced from the scaffolds we use, depending on their stiffness or softness, etc. So here I just want to give you an example that it was shown that when stem cells are seeded on substrates with different elasticity or stiffness, they will differentiate into different types of cells. So if it's a soft substrate, they give rise to like brain. If it's intermediate stiffness, it will be a muscle. If we use a very hard substrate, very stiff substrate, these cells will actually differentiate into bone. So you can see that cells also can sense mechanical properties very well. And then there's also the topography or the architecture of, for example, scaffolds. And here's just one example where the cells were seeded on the same type of substrate with different sizes of pores. And you can see by how the cell behaves here, it takes different shapes, that it actually can sense this different architecture and will change its behavior accordingly. So now this is basically, one more time, the triad of tissue engineering, right? So we use cells, scaffolds, and biological vectors, and I have given you examples of all, to generate tissue-like structures. So now, what have we done, right? So we started somewhere in the 1970s, 1980s, now we are close to 2020. So what significant discoveries were there? We actually covered some of them, right? So we talked about the tissue engineered here. We talked about the embryonic stem cells. We talked about 3D bioprinting. But just to show you what we actually are able to do is that in 2006, we actually engineered our first bladders and successfully implanted them into humans. Here, by the way, this is when the induced pluripotent stem cells were also identified. Then the bladder is obviously a more simple tissue, but we have already shown in the rat that we are able to generate liver tissue in the lab and transplant it into animals, and these livers are actually functional. And recently we were able to do this with lung tissue as well in a large animal model. Right? So there have been some advances, and I just want to give you two examples, and it's quite cool that you already mentioned it. It's the trachea, right? So there have been successful transplantations of tissue engineered tracheas. So in this case, it was actually a trachea from the cadaver was taken and it was decellularized, so only the extracellular matrix scaffold was left, and it was reseeded with the cells of the patient and implanted. And this was successful. The patient is still alive, from what I heard last time, right? And one thing which is quite interesting is that for the trachea, the tissue is in a way special that it's exposed to an air and also liquid interface. So when we were growing, not we, but tissue engineers in general, when these were grown in the lab, the bioreactor was made in a way that there would be an air and a liquid interface so that the forming tissue would be exposed and would get used to both. Similarly, there have been also some successful approaches to tissue engineered urethras, which is basically the tubes which is below the bladder and allows the outflow of urine. So there have been some congenital problems for young patients where this doesn't happen. You can imagine, very painful, it leads to kidney failure or short-term. So here also the patient cells were taken and they were seated on synthetic scaffolds made of PLGA and implanted. And you can see that we were actually able, tissue engineers were actually able to regenerate the urethras. So they actually allowed a normal life for these patients. So we could say that, well, despite 30 years of dedicated research, we are still relying on organ donation, right? So we are still not there. Rather than growing kidneys, livers, hearts, etc. So the question is, what real application are there? And just to point out, 30 years is not a lot for a medical research field. So tissue engineering is still in its baby shoes. And if you think about like that it's really not that old. There have been amazing advances. We have wound dressing, which really promote wound regeneration. We can create heart valves, or we can 3D print scaffolds, which will promote endogenous healing of bone tissue, which especially for craniofacial injuries very important because we want to restore the proper shape of the face. So these tissues can be, so these scaffolds can be printed exactly into the shape which is needed and implanted, and then it serves like a guideline, and the bone can regenerate and the face will basically look as it did before. So since we are already running out of time, I'm just going to tell you what are the challenges and what are the future directions of tissue engineering. We have made huge progress. It's amazing what, if you just think about is where we started and how many people already profit from the advances made in tissue engineering. But right now on the clinic, we are only able to really produce simpler avascular structures. This is partially because 3D bioprinting is still pretty young as well. So it's very hard to make complex tissue structures. If you, for example, look at the liver, you see it's not just a bunch of the same cells, but there are a lot of different cell types which are arranged in a very specific way to form a liver. So we actually still struggle to form this complex tissue structures, although, as for example mentioned in the historical timeline, there are successful animal experiments, and it's a matter of time, and you'll be also able to do that in a human being. The other thing is, the moment we move into a larger construct, we have to make sure that these constructs are well vascularized. Actually, we're pretty good in making small vessels in the lab and also within tissues. So, but once we implant them, they have to reconnect or connect to the surrounding vasculature of the surrounding tissue really fast. Otherwise, our construct will die. So this reconnection is really not easy, and this is also a very active research area. Other issues that have to be tackled are sufficient cell numbers. So I told you that our stem cells proliferate quite a lot, but there are also limitations. So if we grow them too long in the lab, they actually can lose their stem cell potential and they can grow old. There's the issue with the foreign body response. So if we implant scaffold, they are always a foreign body. And our immune system and all the cells of our immune system have had time, millions of years during evolution, to perfect their way of identifying what is their own, so what is owned to our body and what is a foreign material. And the moment they know there's a foreign material, and they always know, they will try to protect the surrounding tissue from this foreign body. So there will always be a foreign body response. The question is just how severe it is, and can it actually be something which then still helps for the foreign body, which is the scaffold, to be accepted. And then immunogenicity, so especially if you don't use the patient's own cells, which is something that also needs to be done because sometimes the patient's cells are just not very healthy, similar to the patient. The moment we move into that, there's obviously always a risk of immune rejection. And this is also a very active research area. So I'm going to stop our lecture here, okay? I'm still open for questions if you would like to ask questions. And yeah, I think what you should know is that if you should decide to go into the tissue engineering and regenerative medicine field, there's still a lot to be done. And this is going to be a research field that will play a huge role in the future. Okay, so thank you so much, and I hope you enjoy the rest of our today's program. And I hope that some of you are going to join us. I'm going to see you maybe next year or the year after. All right, thank you so much.